Blood test. Hematology. Hematology is derived from the Greek hemato, relating to blood, and logi, relating to study. The main purpose of hematology is to study blood and or organs related to the blood. Blood tests are also referred to as hematology tests. Types of blood specimen would be whole blood, serum, and plasma. There are 3 types of blood specimen that are used for analysis: whole blood, serum, and plasma. Whole blood, collected and mixed with an anticoagulant so that it will not clot. Is used for most hematology tests, including blood types, cell count, determination of hormones, and metals. Serum. Serum is the fluid portion of blood that remains after clotting. An anticoagulant is not needed when collecting serum. Clotting begins about 30 to 60 minutes after blood has been drawn, after which the serum can be separated from the sample by the centrifuge, which is a device that separates component of blood based on density by spinning the sample at a very high speed. Serum. Serum is used in many lab tests, mostly blood chemistry and immunology. Unlike plasma, serum does not need fibrinogen and other clotting factors. Plasma. Plasma is the fluid portion of the blood that includes fibrinogen and other clotting factors. Plasma is obtained from the whole blood by first adding an anticoagulant and then centrifuging. Plasma is collected for coagulation studies. It is also used for stat chemistry studies when there is no time to wait for clotting to occur before centrifuging. Remember, it takes 30 to 60 minutes for clotting to start. Common blood tests. CBC, which is known as complete blood count. The most common hematology test is the complete blood count, which is a CBC, which is performed on whole blood. When a CBC is drawn, it measures all blood components in order to fully analyze the patient's blood. White blood count, which is WBC, number of white blood counts, which is the leukocytes, in a sample of known volume, which can identify infections and other conditions. Differential, which is DIFF, classifies and counts the different types of white blood cells as well as abnormalities in red blood cells or platelets. Platelet count, number of platelets in a known volume. Mean platelet count volume, which is MPV, assesses platelet volume and size. Red blood count, which is RBC, number of red blood cells, which is erythrocytes, is in unknown volume. Then the hematocrit, which is HCT, is a percentage of total blood volume in proportion to red blood cells. Hemoglobin, which is HGB, measures the level of hemoglobin in the blood, which determines the oxygen carrying ability. So that test, all of those things, is under complete blood count. So CBC, when someone does a CBC test, which is one of the most common tests, it's the most common hematology test. They're counting the white blood cells, the diff, the platelets, red blood cells, hematocrit, and hemoglobin. Now the next common test is red cell indices. It means corpuscular HGB. I don't remember reading this in school, but anyway, determines the average amount of hemoglobin in the average RBC. It also does MCHC is under the same. So red cell indices would be MCH, MCHC, and MCV, and RDW. So MCHC is the mean corpuscular HGB concentration, which determines the ratio of hemoglobin in a cell as compared to the size of the cell. Mean corpuscular volume, which is MCV, determines the volume of the average red blood cell and assesses morphologic shape-related abnormalities. Red cell distribution width, which is RDW, determine the range of sizes of RBCs. Now the next type of test is coagulation test. Coagulation testing is performed on plasma. These studies are collected in tubes containing an anticoagulant citrate, which preserves the coagulation factors. Coagulation tests, which monitor anticoagulant therapy, are performed on patients with history of strokes, heart attacks, thrombophlebitis, which is blood clots. Drugs to prevent clotting and aid in avoiding a recurrence of the above listed condition are given prior to the test, and the patient clotting coagulation time must be carefully monitored. Clotting disorders such as hemophilia, which is patient blood does not clot, must also be monitored. Common coagulation tests include activated clotting time, activated partial thrombin time, bleeding time, factor activity assays, fibrinogen and fibrin degradation tests, international normalized ratio, prothrombin time, and thrombin clotting time. Common chemistry tests and panels. Most blood chemistry tests are performed on either serum or plasma.